Hello, everyone, and welcome to the IFST webinar, Cultivated Meat, Challenges and Innovation in the Sector, organized by the IFST Food Processing Special Interest Group, or SIG for short. My name is Kavya. I am the Vice Chair of this SIG and a Food Innovation Technologist at the University of Nottingham's Food Innovation Centre. We're a commercial arm of the university, supporting businesses with technical and scientific support across the UK. Cellular agriculture is a hot topic across the world and approved for sale in some markets. The IFST SIG has put together this webinar to give food science and technology professionals an overview of the production process of cultivated meat. For cellular agriculture to become a meaningful contributor to current protein requirements, the technology and conversation must move beyond a few key players and involve those who currently make our food systems and supply chains work. Various providers across the supply chain need to come together in order for a manufacturer to have an end product. And through this webinar, we hope to give attendees a holistic introductory view of the manufacturing process of cultivated meat. Some general housekeeping. This webinar is being recorded and will be uploaded to the IFST's webinar hub. Feel free to make comments and ask questions throughout the webinar using the Q&A button at the bottom of your screen. You can also upvote other people's questions by clicking the like button next to their question. And there will be a Q&A session after the presentations where we will respond to as many of these as possible. I would like to introduce our speakers. Our first speaker today is Professor Ramiro Alberio, Professor of Developmental Biology at the University of Nottingham and co-founder of Pluricells, a licensable technology providing embryonic sheep, cattle, and pig stem cells to the industry. He graduated as a veterinarian from La Plata University in Argentina and gained his PhD from the University of Munich. His laboratory investigates the molecular mechanisms governing acquisition of cellular identity during embryo development, and he used this knowledge to found Pluricells, which supplies high-quality livestock embryonic stem cells, which have broad applications in biotechnology to the cultivated meat industry. He is the director of the Center of Large Animal Biotechnology and deputy research director of the School of Biosciences. We are also joined by Kai Linton, co-founder and chief executive officer of Multis Biotechnology Limited. Kai has a master's in molecular bioengineering from Imperial College London and combines technical knowledge in tissue engineering and data science with management experience. His motivation to create a sustainable future using biotechnology led him to co-found Multis to shorten routes to market and lower entry barriers into the cultivated meat industry. Multis makes faster and cheaper growth media that scales, accelerating the adoption of cellular agriculture. The company's growth media formulations and ingredients are the building blocks of cellular agriculture and enable the affordable at-scale production of real animal products, including meat, dairy, and leather, and more, using cells instead of animals. For more information on our speakers' biographies, please look on the IFST website. And with that, I will hand over to you, Ramiro, to talk us through cell line.
So I would like to talk to you today about the work that we're doing in our lab to develop cell lines that are, in our opinion, very useful for developing this technology underpinning cultivated meat sector, a sector that has only been developed over the past 6 or 7 years across the world, and where there is a lot of opportunities for innovation. So our lab is a developmental biology laboratory, so that means that we work with embryos and stem cells for multiple applications. But when the topic of cultivated meat appeared through contacts and through various meetings about 5 or 6 years ago, we became very interested in explore whether our cells would be of any use to this industry. And the point of cultivated meat as a solution to the problem of the demand of meat globally was really posed a few years ago, about 2013 or thereabouts, a bit earlier. And the first example was presented by Mark Post, an engineer from Holland, who produced the very first burger made of cells grown in the lab. And in essence, at the time, it was very visionary. It was very provocative, and people took this as a almost as a joke, really. But since then, there has been a lot of effort in trying to make this process, which is summarized in this diagram here, as viable as possible financially, but also that results in a product that is tasty, nutritious, and sustainable, particularly. And so, in a nutshell, this is what the process entails. So we have an animal source of cells. Typically, these are taken from living animals, but it could be from various sources. The cells are then expanded in the lab at large quantities. And then there are two options, either to produce an unstructured product, which would be more like a mince that could be used for meatballs or sausages or burgers, etc. Or the alternative is to make a structured product that would be significantly more expensive. But the point of this is that ultimately what we're trying to achieve with this type of technology is the reduction in the impact in producing um, meat from animals by reducing fundamentally the carbon footprint, particularly with beef, where it is what it has the most impact globally, and the use of land. The demand of meat globally is predicted to increase by 50% in the coming 2 decades. And so we have a real challenge in our hands in trying to supply the demand in a sustainable way. So we believe that cultured meat could offer an opportunity for satisfying this demand. Now, if we look into the details of how this can be made and the type of sources of cells, which is what we focused on, we have this in this diagram, which looks quite busy, but I will break it down for you in simpler ways. There are three main sources of cells that can be used or have traditionally been used for this process. And in essence, is the source of cells that are the progenitors of what constitutes meat, which is basically muscle cells, fat cells, and connective tissue cells. So typically, as highlighted in this red box, you can see cells from the muscle biopsies or tissue biopsies that contain different type of cells. These cells can be expanded through different means by immortalization or just expansion of the stem cells. And these cells can be then introduced into bioreactors and expanded in large quantities to produce the final product. An alternative option, this is what most companies are really using or have traditionally been using since the inception of these products. A more recent development has been the formation, the creation of embryonic stem cells, where we have taken a major part in the development of this technology, which is basically taking the cells before the cells are committed to any tissue and the blastocyst in the early embryo. This is a pre-implantation embryo, where the cells can be isolated and expanded. And these cells retain capacity to make any cell type. That means that you can instruct the cell to give rise to muscle fat or fibroblasts as at will, depending on the protocol that you choose. And another alternative is the third option is to induce pluripotent stem cells, which is taking a biopsy of cells of an animal and then reprogram them using chemical approaches in the lab. So these are the three main sources of cells available for this technology, and assessing which one is the most cost-effective and more efficient will be really what will make a big difference to this industry in making it more viable. So in our case, I will briefly outline how we develop these cell lines that are highly potent in their proliferation in vitro. They grow under conditions that are suitable for as a food product because they are under chemically defined conditions. That means that there are no undefined product used to grow the cells, no animal-derived product. They don't grow using any feeder layers, typically coming from other species. And they can also grow long-term, which means long-term self-renewal or long-term expansion is essential when you have a manufacturing process ahead of you. They are also karyotypically stable, which means that the cells will not undergo abnormal phenotypes over time. And they maintain their features of pluripotency, which defines their characteristic differentiation potential. And they also map, this diagram here at the bottom is showing you a spread of cells with different colors, and the green cells represent the stem cells that we have isolated, and the orange and blue cells represent the equivalent cells inside an embryo. So these cells match quite well the cells inside an embryo, which means that by having cells that have very similar characteristics to the cells in the embryo, we can then induce the differentiation following the normal path of differentiation that muscle cells or fat cells will take in vivo. So having these cells, then we tried the same protocol in other species. We are interested in also making obviously lamb and beef as a cell type for consumption. And these protocols apply to all three major livestock species, therefore. And they can be applied very efficiently. Now, when we take these cells, we can also induce the differentiation to muscle. This is a little movie showing you here the contraction of the skeletal muscle cells after a 10 or 15-day differentiation process. You can see that they are contracting like a muscle cell. And if we look for markers of differentiation, we can see these immunofluorescent images showing typical proteins expressed in the muscle lineage. So these cells can undergo terminal differentiation, for instance, to the muscle lineage, depending on the protocol that we use. So that means that we can produce muscle progenitors and muscle and myotubes very efficiently. Now, if we contrast the different sources of cells, there are some advantages and disadvantages of using different types of cells, and this will be probably of the discussion later, but I just wanted to highlight some key features of the different types of cells. So the origin of the cell is a very important aspect from the point of view of safety. In all cases, we can have a good traceability. We know what animals are going to give us the biopsies. The use of antibiotic typically is required for when you obtain a biopsy from a tissue, from an animal. In the case of pluripotent stem cells, that's not required because the cells are obtained in a sterile environment, and that's not necessary. The differentiation potential of embryonic stem cells is very high because the cells maintain the undifferentiated characteristics over a long time. That can be more variable depending on the source of cells from the somatic tissues or the biopsies that we can access. And the proliferation is also unlimited. So embryonic stem cells are very rapidly dividing cells compared to other somatic cells, which are much slower sometimes, depending on, again, on the source of the cells. They have long-term stability for somatic cells that can be quite variable. And of course, if you undergo immortalization, that can the cells can accrue some chromosomal abnormalities that might impact the differentiation potential eventually. And of course, we have reproducibility in embryonic stem cells. The protocols are well established and they're highly efficient in their derivation, and of course, they're commercially available. So we established pluricells as a platform for commercialization of these cells about 3 years ago, and basically we comply with some of the current standards for food grade cell lines. Those we can discuss them later, but basically haven't been completely defined by the regulator. But we try to, we do know what to expect from what the standard food standard cell would require, and we comply with some of those already. And these cell lines can be banked from multiple breeds, for example, and it gives plenty of choice to the basically to the manufacturer in the type of cells that they can acquire. They have rapid proliferation and stability, which is what they require when they're trying to differentiate cells in large quantities. Now, in terms of what we're doing next, we're developing the cell lines, but we're very interested in understanding what are the bottlenecks for this industry. And clearly, there are many issues, which are some are highlighted here. Like, for example, the sourcing of cells is a very important and critical aspect that needs to be developed going forward. We need to reduce the production cost. For example, the expansion of the cells at the moment is a very expensive aspect of the process. Improving the differentiation of the cells, so inducing the cells to differentiate efficiently into muscle and fat or connective tissue is critical to ensure that the product is always very reproducible. Scale up is also a major bottleneck. Producing metric tons of meat in this type of approach has never been done, and we are still developing the bioreactors and the capacity globally to be able to match what can be done with living animals. I think we have a massive challenge ahead of us in trying to scale up this industry. And of course, it's important to not only produce a product that will be on price parity with current products, but also that is a product that potentially offers nutritional advantages over current sources of meat. We can produce a distinct product that might be hybrid and contains different proportions of proteins and of animal origin and vegetal origin, so that the product can be actually healthier for the consumer. And of course, sustainability is a key aspect, a key driver for this technology. We need to ensure that this is a sustainable way of making meat. So all of these are big questions, big areas that are needing a lot of collaboration and work. And I wanted to highlight that as a result of all these questions and the expertise that exists in different areas of biotechnology and academia and industry, that a supply chain is developing as a result of this interest. There are many companies that have specialized in different aspects like cell lines, culture media, scaffolding, bioprocessing, etc., that all of them are contributing to making this a more sustainable and very well designed industry that will have basically experts contributing to different aspects. It will basically like the car industry developed over the past 50 years. Hopefully the same will take place with the cultured meat. So with this, I'd like to finish. I think I've spoken for quite a few minutes now, and just like to finish by acknowledging the people in my lab. We have a cellular agriculture lab here in Nottingham, working within the University of Nottingham, and my key collaborators are listed here: Doris, Nathan, and Terry, who are basically primarily growing our cells, and Charlotte, Luke, and Kathy, they are primarily involved in the differentiation of our cells. We collaborate with multiple academics. These are some listed here, more recent ones from people in different parts of the world that are developing similar technologies. And of course, acknowledge our funder, the BBSRC, who's been a great supporter of this work. So with this, I'd like to finish, and I will probably now pass on to Kai to continue his work, and we'll take questions afterwards. Kai, all yours. So I will stop sharing.
Yeah, thanks.
And mute myself. Thank you.
Just share my slides now. Okay, great to be here, everyone. My name's Kai. I'm the co-founder and CEO of Maltus. We're a company that's really trying to grow the sustainable bioeconomy so that companies can produce better products for people and the planet. We started off in 2020. My background's in bioengineering. We began at Imperial College London, joining the IndieBio program, which is one of the largest life science accelerator programs in the world. Our focus then was really about helping companies optimize the feedstocks that they use in biomanufacturing, so otherwise called the growth media, that provides all the food that cells need to grow in these manufacturing processes. And so we had our first product launch in 2021, which was a serum-free formulation designed for the cultivated meat industry. We then built out our capabilities, so ISO certified manufacturing capacity, and then a pilot facility that followed, closed in total now about £13 million. And in 2024, brought our high throughput media optimization platform online, which is what we're using now to develop our next generation formulations. Ramiro talked a little bit about this, but really the livestock industry has an outsized impact on the planet. Since the 1960s, we've increased our agricultural efficiency by about 3 times. And currently, if we continue that kind of improvement, so continue improving at the same rate, we'll still need about 2 Indias worth of land to meet the increasing demand over the next 25 years. So quite clearly, we need quite a radically different and more sustainable, more efficient production system, because the current system is consuming the planet in terms of land use, in terms of antibiotic use, diversity, as well as the greenhouse gas emissions that it causes. So cultivated meat offers a solution for this, but today it's just far too expensive. And one of the key reasons it's so expensive is that the feedstock that companies use just isn't designed for the food industry. Up to 80% of production costs comes from the feedstock, but the tools we have to lower that cost, optimize those formulations, are just far too slow and expensive to help this industry get from lab to market. And so this media is a big blocker for the industry right now, and really risks many of these products succeeding in the market or even reaching the market at all. And this is because the traditional way of developing new formulations is very slow and expensive. It often takes companies 2 to 4 years through a very manual process of trial and error, often resulting in formulations that are still very expensive in and of themselves, and using ingredients that are just not compliant and suitable for the food industry, both in terms of regulatory compliance, but also in terms of their availability at scale. And so at Maltus, we've brought a new approach to this problem. We're rapidly increasing the rate of new product development, bringing down the optimization process from years to months. And that's enabled by an automated platform that allows us to capture far more data and use that data more efficiently to find these optimized formulations. This allows us to bring down the cost of media formulations much lower than the gold standards today, using ingredients that are completely animal component free and compliant with regulations in the food industry. This is enabled by what we call our media platform. It's our way of optimizing media formulations through a combination of robotics and machine learning. And so through automating our experiments, we can run about 100 times more experiments per scientist enabled by robotic automation. And from those experiments, we're collecting a lot more high quality data. And we can run that data through different machine learning algorithms to extract valuable insights into how these cells are growing and behaving in different combinations of ingredients. And what we start to do is build this iterative closed loop formulation design process, where we're then using machine learning models to take that data and design the next experiment that feeds into our robotic process, collects more data, analyzes it, and designs the next one. Because we've automated this process, it allows us to move very quickly from a set of ingredients to optimize formulations. And so to give you a bit of a case study, last year we had 2 scientists working on a new formulation. This was for bovine fibroblast cells. So these are cells that haven't been industrialized before. There aren't off-the-shelf media formulations readily available. And so we had 2 scientists work on this problem. They were optimizing about 30 ingredients in parallel, including 5 what we call complex ingredients, so plant-derived multifunctional ingredients. And over the course of 4 experiments, they tested several thousand different combinations of those ingredients to land upon many formulations that are now outperforming high serum concentration growth media at much lower cost. And so this is just showing on the cells that we're working with, we had several formulations outperforming FBS, and we've now tested that on other cell types as well to show that we can sustain growth over a wide range of cell types in this space, including fibroblasts, which are kind of skin or connective tissue cells, as well as muscle cells. We've done this as well on these IBSCs, which are quite commonly used now in cultivated meat research. And so as a company, we do 3 key things. We help really companies at the R&D to pilot scale develop and source their growth media. We have off-the-shelf formulations like the one that I just showed you. These are ready to grow. We've formulated them generically for common cell types in this space. But we do a lot of work with companies that want custom formulations. This is through a development service where companies come to us with their cells and their objectives, and we find the right combination of ingredients to maximize productivity, functionality, and lower the cost. And then we've built out the manufacturing capacity with food safe certifications so that once we have a formulation, we can help companies get it on their journey from the lab to market. And so in terms of working with us, we have several off-the-shelf formulations, as well as the services that we'd love to speak to companies about who specialize in formulation optimization, but we're also venture-backed. And so if there's interest from corporate partners or investors that might want to discuss our funding roadmap or just sign up to the updates over the next few years, then please get in touch. Thank you very much.
Lovely. Well, thank you, Ramiro, and thank you, Kai, for very interesting and engaging presentations. We'll now move on to the Q&A session. So just a reminder, you can submit your questions at the bottom of your screen using the Q&A button. And we've got a couple of questions actually to start us off. So I think, Kai, on the back of your presentation, I'll just ask you the first question, which is, so many startups begin with a vision to solve a problem, and yours was to tackle media costs. So what you mentioned in your presentation that 80% of that cost is, in the supply chain is usually media. So what makes media so expensive?
Yeah, good question. There are two key drivers in the cost of media. One's the ingredients themselves. The second is the efficiency of the formulation. And so the way that we tackle those is we do a lot of work trying to build out an ingredient library that's qualified for this industry. That means often moving from biopharmaceutical grade suppliers to food grade suppliers, from animal derived ingredients to plant derived ingredients, and making sure that those ingredients are not only cost effective, but also scalable and functional in the cells that we're working with. The second part of that is then finding those combinations. What is the formulation that's actually going to grow these cells most productively? And so where you can have cheap ingredients and efficient formulations, that's what will get you to lower media costs. But today, a lot of the formulations are just not designed for cultivated meat cell types and are being used in a different industry, the biopharmaceutical industry, and that makes them very expensive.
Okay, thank you. I guess both of you touched on the food grade, aspect of cultivated meat production. So, Ramiro, would you be able to tell us what makes a cell line food grade exactly? Because you also spoke of traceability, and that's one of the things that Pluricells assures. Yeah, just what makes a cell line food grade?
I mean, we have to bear in mind that the regulator hasn't really issued a very clear guidance as to what food grade is. They are still working through the process of determining what that is. But we can guess a number of things that are going to be required, and this is based on current regulation for food products. So clearly, the origin of the sample or the origin of the cells used for this is important because you need to determine that the animal was healthy at the time when it was obtained, or was it a cadaver, or was it a living animal, for example. So the origin is important. And also particularly in this world where animals move around, diseases move around, and ensure that the origin is from a safe place. Secondly, we have then the things relating to pathogens. So normally abattoirs and places where meat is manufactured, processed, we check for pathogens, and so we will have to ensure that we don't have pathogens in our products. So these are quite the obvious things that one can expect the regulator to be interested in having at least the same quality of product, or if not better. But then we have the aspects relating to the manufacturing itself, which in this case it will be relating to aspects of the bioprocessing. So the expansion of the cells, the type of media components, as Kai was saying, if these are animal-derived free components, that makes them definitely safer than if they are not. And the use of fetal calf serum has been an issue for a number of decades, which is a typical component added to the culture medium. That would not be acceptable as a food grade product because of the requirements to show origin and also batch variability. But then finally, regarding the bioprocessing itself, we need to think that we need to produce these products rather than in an animal, in vessels. And vessels, we need to ensure that these vessels don't contain any contaminants introduced as a result of the manufacturing process itself. So there will be requirements for demonstrating lack of allergenicity, for example, with some of the compounds that are produced by the cells when they are largely expanded, or for example, trace minerals or trace plastics, for example, or anything to do with the bioprocessing that is novel to this industry. So this, as I said, it hasn't really been legislated or carefully issued as a guidance from the regulator, but the regulator is working on this. In fact, we are contributing to this regulation. We are, as academics, we've been invited to participate in some of these events where you're trying to identify what the hazards are and what the risks of these hazards are in the final product. So the guidance is being produced as we speak, but we can really have educated guesses as to what they are, and some of them is the ones I just outlined.
Thank you. We've got a related question, which I think you touched upon some of it, like pathogens and allergenicity and the risk that poses to health. But are there any particular risks to public health from using some of the stem cell lines that you mentioned? And that question is specifically for Ramiro. For example, could there be any contamination due to animal health issues?
No, so that would apply to other cell lines too. So long as you demonstrate that the cells that are obtained from those biopsies or from the embryo samples are coming from healthy animals, and the cells themselves can be screened against pathogens. So there are multiple batteries of kits that you can use to determine the level of sterility of your cultures. These are routinely done in our labs. We can demonstrate that they're totally, basically, pathogen free. So from the point of view of contaminants, I think that's a relatively easy way to test that. Also, because if there were any pathogens in the culture, it would actually destroy the culture probably over time. So it will result in no growth of the cells.
Okay, thank you. We've got actually quite a few questions. So the next one is, would you say the UK is ahead in this field compared to other countries? And maybe we'll start with Kai, and then Ramiro, you can offer your opinion on this.
So in some aspects, the UK, yes, is quite a leader in this space. We have a very rich ecosystem, as Ramiro showed, of not only cultivated meat producers, but also enabling technology companies. We're starting to build quite a strong academic base, although this is certainly catching up. We have Karma, which is a project around cultivated meat manufacturing out of Bath University. We have the National Alternative Protein Innovation Center, which is a national consortium, but led out of Leeds University, and the Basel Center for Sustainable Protein Production at Imperial College London. So these are world-leading centers now with a leading amount of government investment and support. Where we're lagging, though, certainly is around infrastructure. We don't have the same infrastructure as other companies, other countries. And whilst the UK government has invested a fair amount in cultivated meat, not as much as places like the Netherlands or Singapore. And we also are in some ways ahead, but not leading in private funding as well. We have the second highest number of companies in this space, but in terms of venture capital investment, we're behind both the US and Israel. And so there's a need for that growth capital as well in the UK to really bring these products to market, which, as always, quite good at the early stage stuff, but are struggling to get these products to market.
Yeah, I mean, I agree everything that Kai says. I agree, and the only thing to mention, perhaps in addition to that, would really help the UK position itself as a leader, is to have regulated products. And now we've recently seen the approval of pet food, made using this process, and that will really attract a lot of investment in this country, because one can see that the regulator is very interested, or at least is basically doing its due diligence and getting these things through the system. So I think from the point of view of investment in the area, you need to see that the regulator is really also part of the solution, and that's very encouraging, because some other places like the EU is a bit more lagging behind right now on this. The US is ahead of us, so is Singapore, but we are there. We are in the top three areas in globally that are really getting these products through approval.
Great, thank you. The next question we've got is, do you see cultivated meat taking over from farmed meat, or what balance do you anticipate? And we can start with Ramiro, maybe, because you sit on a campus which is, farm to fork. So you've got a spectrum. So
Yeah, so I think it will be a mistake to try to put these two sources of meat as competing sources. I think we have outlined very clearly that the demand for meat will increase by more than 50% in the coming decades. So clearly the conventional meat production cannot satisfy this demand without destroying the planet. So we need to come up with an alternative way of doing it. So I would say, particularly in my slide where I showed you the unstructured versus structured products, I can see this technology really playing a massive part in unstructured products, which is what most people actually consume on a daily basis. The steak is maybe more the once a week, the sausages and the burgers are maybe twice a week or more. So I think we are trying to complement this industry by offering products that are typically sub-premium, because the products that give rise to these products I just mentioned are not really the prime steaks that are being minced. It's just the rest of the animal. So we think that this can complement very well the regular industry. And in addition to that, it can represent a source of income for farmers, because if we think about the sources of sales that we just mentioned, they have to come from somewhere. And so for farmers, this could also represent an additional source of income if they were to engage with this type of novelty in the way they produce the meat.
Great.
Yeah, I'd agree with that. I think the challenge is over the next 25 years and beyond, how do we create a food system that's a lot more productive but doesn't use as many resources? And so we need more productive meat systems. The UK already uses about 70% of its land for farming, but imports about 45% of its food. And so I think farming can for sure live alongside, and it will live alongside, cultivated meat. We're a very small and nascent industry today. But the problem is not farming versus cultivated meat. It's our food system versus growing demand and sustainability.
Yeah, it's a very interesting question. We've got another one, a good question after this, but most of this projected demand is going to come from poultry. And we see that maybe pork meat and beef is maybe in second or third place, but it's really poultry that's driving this demand, especially in developing economies. The next question we have kind of ties into that, which is, are you or anyone in the UK working on cultivated poultry or fish for that matter?
We're doing some projects with fish companies, not based in the UK though. Poultry, not at the moment, but it's certainly very popular. The challenge there is chicken and fish are quite efficient. And so, yes, there are aspects of animal ethics and certainly overfishing that are problems too. But in terms of the environmental impact, fish and poultry are quite productive, and so are difficult to compete with on price. Although fish, you have the argument for sure in terms of sushi, etc., which is quite attractive for lots of companies.
Yeah, Ramiro, do you want to?
Yeah, so I'm not aware of companies working on poultry in the UK. I believe in the US there are a couple, but as Kai said, I think there is an issue of the effort that takes to do this, and what will be the benefit really to the final in terms of financial benefit, but also the sustainability benefit. I think anything to do with a large animal like a pig or cow or a ruminants, particularly because of the greenhouse gas emissions, I think that's what the major advantages to the planet and to the food system, to have a sustainable source of meat. Poultry is kind of a little less critical, I would say. But I'm not saying that it shouldn't be done, but for all the other aspects relating to production system, but it probably represents less of an advantage to work on chicken.
Great, thank you for answering those. There are two questions around consumer acceptance and perception. So, what are the main consumer challenges that you foresee, in terms of how it's perceived? And how would you navigate these by collaborating with different stakeholders? What do you think needs to be done?
I mean, Kai, you want to go first?
Sure, I can share some thoughts. I think people primarily make buying decisions based on price, convenience, and taste. And there's lots of evidence to support that. However, cultivated meat is a new food, and that creates some concern for people. It's processed in a new way. It's unfamiliar. in terms of how we support that consumer acceptance, one important part is trust in the regulatory system. Is the Food Standards Authority not only making sure that these food products are safe, which they are, that's their job, but also building trust in the process so that when people buy these products, they are confident that they're safe to eat. The other part is around transparency. We can talk a lot about our production process. We can show people the production process, and that starts to build trust with consumers. That's often not a thing that you'd get in industrial farming processes. And so I think transparency and working with robust safety regulations is one way to build that initial trust, and then get products that taste good and don't cost so much on the market in front of people to try.
And also, it's important to mention that food obviously is something very culturally of great importance, and so creating this in a different way to what convention dictates can create some doubts to people. So that's why I think I emphasize the type of product that this industry is aiming at right now, which are probably less linked to a specific tradition, if you like. Yeah, eating your steak in a particular way or sourcing the steak in a particular way. Sausages, burgers, and meatballs are typically very convenient sources of meat that are fairly cheap and tasty. And so we are not competing with those other aspects relating to food, which is more like the cultural aspects. It's more of a convenient product that will be available. And with the advantages, in my view, and that's what interesting consumers are very into. I participate in several consumer groups discussions, is the fact that this could be actually be made as a healthier product. So you will be eating healthier meatballs because there will be a hybrid content of protein coming from different sources, for example. So you still get your animal protein quality, high quality animal protein, but you're also blending it with some pea protein or soya protein or other sorts of protein are also healthier for your gut and so for your so I think this is a very attractive proposition for the consumer in that you're offering potentially a better product and hopefully on price parity and taste parity with conventional products.
Okay, thank you. What is the next question we've got is what is the approximate timeline when we can see cultivated meat on UK shop shelves? What are the three main obstacles of stopping this technology and its wide adoption?
It's available today if you'll eat dog food. So it's cultivated meat. Dog food has been approved. You can buy that today. There are several companies, I think at least 5 companies that are in the regulatory approval process now in the UK. And the UK just began a new regulatory sandbox to work with industry and other stakeholders to build more proactive and efficient approval processes, not kind of lowering the bar of what we need to achieve, but equipping the regulators with the tools that they need to assess these new technologies. So that will help many more products get to the market. I think in the next 5 years, we'll see several cultivated meat products in restaurants, at least for humans to eat. In terms of barriers, cost is a major one. Scale is another. That's mostly a financial barrier, and it's a difficult time right now to get the kind of money you need to build a big new factory to make cultivated meat.
Ramiro?
Yeah, I know. I echo this point. I think those are the main ones, and so the regulator will play a massive role in promoting this, or the curiosity by the consumer, because once you know that the FSA has approved a product, and clearly obviously is safe and it's been demonstrated to be fine, I think the curiosity will definitely drive massive demand because. I think it's a very interesting proposition, and particularly in the innovative ways in which companies are thinking to present the product. I think it will attract a large group of people in the population, and I think definitely demand will outstrip because the problem is actually production. So you cannot just launch a product and then you run out of stock within weeks because you cannot match the demand. I think that's a serious problem. I think scale up is a serious problem for this industry right now, and it's a bottleneck really because maybe the regulator will eventually get to approve these things, but we're not going to be able to produce enough for the markets that we are aiming for.
Are there any health risks, to the best of your knowledge, associated with more long-term consumption of cultivated meat that you know of? And Ramiro, do you want to take this?
I mean, we do highlight in the safety aspects that I was mentioning before. There are aspects that need to be evaluated. First of all, it's important to highlight that we are producing meat. We're not producing a meat analog. It's actually meat. So it has fat, it has muscle cells, and it has connective tissue. So on that basis, it's the same as having it from an animal. It's just it comes from a bioreactor, but it's the same. It's not an analogous substance, so it's the same. The only one of the aspects for particularly to consider is allergenicity, for example. These are things that can be tested. Are we, as a result of the proliferation of cells, creating cells that might have mutated to a point that they're producing proteins that are normally not there? And these are things that can be tested for example. So allergenicity, I would say, is one of the things I would be more interested in testing from the point of view of safety. I cannot think of many other things. Considering bioprocessing is being taken into account in the sense of that there are no contaminants, etc. But from the point of view of the safety of the cell itself, the product itself, I think it's allergenicity one of the main topics I think I would focus on.
You also mentioned mutation. So we've got another question, which is there any risk of cell mutation when growing the cells? And
Yes, of course, mutation happens all the time in every cell in the body. That anytime that it divides, it will acquire a mutation, a single point mutation, typically irrelevant to the cell's viability. Spotting those in large bioreactors can be done by sampling and by routine checking and by making sure that you have the right media and right conditions. Those mutations do not necessarily cause any impact in the final product until they do. And when they do, you will realize that because you will have screens. So mutations is something that regularly is tested for when you're growing cells at scale. And that's something that can or cannot have an impact. It depends on the mutation. Because it's a random event, it's hard to account for. Whether it impacts the final product from the safety point of view, I don't have serious concerns, except if you end up with a mutation that causes a protein to misfold and it causes allergenicity, for example. So that's the type of relationship to allergenicity that I was trying to draw the attention to. But apart from that, typically, if there is a mutation that is either lethal, it will kill the cell, so they're not going to be a product, or they won't differentiate into their tissue of interest, and therefore you end up having a very inefficient system of differentiation. Your product is very bad, so you don't commercialize it. So these are things that can be tested before the product reaches the market, basically.
great, thank you. Kai, do you want to add anything to those last two questions?
No, I think Ramiro covered that quite well. Cultivated meat is a new manufacturing process, so it's scrutinized very heavily from a safety aspect. Every part of the production process has quality controls. Every cell is screened at the genetic level repeatedly. Every ingredient in the growth media is considered individually. Because it's a new production process, we can use kind of modern food safety scrutiny on it. Lots of foods we eat today don't have the same scrutiny. There are mutations in animals, of course, every day as well. So cultivated meat from that aspect benefits from much higher level of safety scrutiny.
There are a couple of things that people keep talking about when they talk about the risks of cultivated meat, and sometimes they refer to the risk of the excessive use of maybe antibiotics, a risk of antibiotic resistance. There are metabolic byproducts, growth factors that are in far higher concentrations in, let's say, in the media than they would be in conventional meat tissue. So do you have any insight into how this could be controlled? And do you want to start with Kai?
Sure. Yeah, I can talk a bit about this. In terms of antibiotic use, so yes, these are used perhaps in R&D, research, kind of early stage product development, but in large scale biomanufacturing processes, by and large, you can run them without antibiotics. It's about controlling the sterility of that growth environment. And in a very standardized production process, that's absolutely possible and has been done before. So there's not really a major need and already an expectation that at scale cultivated meat products will use antibiotics, certainly not in the quantities that are used today in industrial farming. In terms of metabolites and media, again, these things get screened and scrutinized at the safety level as part of quality control to look for the presence of these components. We recently did some work funded by the UK government to develop some new methods for detecting the presence of residual growth factors in the final meat products and comparing that to conventional meat. I won't share all the results there, but just to say that what we detected was much lower than conventional meat.
Thank you, Ramiro.
Yeah, you have to remember also that this product will eventually be consumed and cooked, first of all. So many of these products, if they are bio products like a growth factor, for example, it will denature probably during the cooking process unless you're eating it raw. You're not going to be exposed to the very vast amounts of it. There is a lot of clearance, of course, during the process of washing, the media off and the blending and manufacturing. And finally, the digestive system that we have denatures everything pretty much. The acid in the stomach will. So it's very, very unlikely that we are being exposed to vast amounts as suggested of this growth factor, which I use in microgram levels. We're talking very, very minute amounts of exposure. So as Kai said, I think there are ways to test for residues of any of this. And these are part of the test that FSA is taking as part of the assessment. So companies are due to produce reports on these things, and these are valid points. But as a biologist, I would say I'm not overly concerned about those. I would be more concerned about, let's say, accumulation of microplastics if the bioprocessing plant has plastic tubing or whatever, more than actually biologicals. Biologicals are used in very, very small amounts of this to cause a risk to a consumer that may be eating, I don't know, 15 burgers a week. I very much doubt that this represents more of a health risk than current type of sources of meatballs or burgers.
Okay, thank you. We've got time for two more questions. So I'm going to pick up on one because the intention of this webinar was to give food science and technology professionals a kind of overview of this because it is ultimately food that we are looking to eat and enjoy. Would the typical manufacture of cultivated meat follow the same HACCP principles as, in quotes, normal food? And what information do you think the average food technologist would need to learn or look at when working with this food category? Ramiro, do you want to go first and then Kai?
I'm not very familiar with what the HACCP principles are, apologies, but I assume if it's the principle for normal food for health assessment and risk assessment, yes, probably yes. There will be additional ones though, due to the way in which the cells are put through a bioreactor. So the pathogen side, all the source of or origin of the cells will be very similar to maybe when you're having animal passport for an animal going to an abattoir. But then you have all the aspects related to the manufacturing. And this is the type of guidance that FSA is issuing or trying to come up with to offer the manufacturers a clear roadmap as to what they're supposed to produce as evidence that this product is perfectly safe.
Okay, so in terms of the microbiological risks, especially, how would you say that this differs from its meat count? I mean, it's live animal.
No, I don't think it's different because you will be looking for those. It's just that you won't have certain pathogens. Like, for example, in pig, you look for parasites. Those parasites will never be found in this type of meat because the parasite's life cycle is incompatible with the manufacturing process. So trichinella, for instance, is one of the typical things you see in laboratories being checked in pigs. That will never happen here because you never have trichinella in this system. So as an example, some pathogens don't have an opportunity to be present in these products. And because it's a bioreactors and they're sterile by definition, they will never have the content of microbiological content that a regular abattoir or regular piece of meat will have.
Thank you for answering that. Kai, do you have anything to add to this?
I agree with that. We have FSSC 22000 certification, kind of HACCP and ISO 22000 together. in our pilot manufacturing facility. It's a food safe manufacturing standard. But as Ramiro said, these cells are quite sensitive. They don't have an immune system kind of in and of themselves. And so the sterility standard actually needs to be much higher than traditional food production processes.
Thank you. So the last question we have, and for all the students joining us on this call, is what degree or degrees would you say are most relevant to a job in cultivating lab-grown meat? And perhaps I'll start with Kai and then Ramiro, because you both come from very different, startup versus academia, and I'm sure that they'll be quite different. So Kai, do you want to take this question first?
I would say everything. You need, scientists in the lab developing new technologies. You need industrial engineers to build out and operate the manufacturing processes. You need people that are good with policy to work with governments and stakeholders to help get these products to market. You need people that can market a product so that it looks good for a consumer. This is a new food category. It needs all kinds of skills to be successful. So yeah, I think there's a place for everybody in this industry.
Ramiro?
And there is no single dedicated course that caters for this type of route of work, but you can see a lot of people translating from the regenerative medicine field, for example, into this area because people have invested a lot of decades into, for example, making muscle for curing human disease. And so much of that knowledge can transfer very well into this, in this type of technology. So anyone working in biotechnology degrees, biology degrees, animal science degrees, everyone in any of the STEM areas, to be honest, including mathematical modelers, can be very useful for some of the understanding of these processes. So there is a vast number of jobs out there that are for people that are very passionate about the planet and coming up with this disruptive technology that can really solve a big problem. So I think this is very, very open to a number of skills that are currently scattered in different fields that can contribute to making this a much more efficient process, really.
Great, thank you. I think we'll take the last question now, which is an interesting one on hybrid meat. So, Ramiro, you said that the scale up is going to be a challenge. Even if this gets to the shelf, then meeting that demand. Some countries are following a hybrid approach. I think Singapore is. So, do you see the market as being more of a hybrid meat sort of market for this particular, category?
I think initially I see it simply from a pragmatic point of view. It's very difficult to produce a sausage that is made of 85% or 95% of this type of, and sausages typically don't have that much meat anyway. So they have maybe 40-50% content of meat. For those that are not familiar with that, I mean, sausages are typically not made purely of meat or fat. So hybrid products offer the alternative that they can also offer this additional benefits we mentioned before about health benefits and mixing proteins from different sources, plus the taste that the animal fat provides and the sensation in your mouth that actually you're eating piece of meat is very important for that consumer experience. But at the same time, you're eating a healthy amount of protein coming from different sources. So I think it has an interesting commercial value to offer something a bit distinctive. And it doesn't compete necessarily with conventional meat, but offers something different for a public that might be more mindful of the nutritional aspects of this meat. So I think there is definitely scope for that. And from the point of view of sourcing that, making hybrid products that have maybe 20% animal protein rather than 60%, well, it makes it much more efficient.
Yeah. Well, I'm afraid we're going to have to stop there because we're out of time. Thank you very much, Kai, and thank you very much, Ramiro, for agreeing to join us on this webinar today. And thank you to all of you who attended. If we didn't manage to answer your questions, and I can see that there are still a couple of them in the Q&A box, please contact our speakers directly. Their details will be sent with the recording and the certificate of attendance. We will share a recording of the webinar, including the Q&A session, to everyone who attended today via email and on the IFST webinar hub. Before you log off, please do fill in our short survey, which will launch after the webinar ends. Thank you again to everyone, and thank you to our speakers. Bye-bye.
Thank you very much, Javier. See you soon.